Hello, everyone, and welcome to the latest installment in Contract Pharma's webinar series, Biopharmaceutical Process Characterization. I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by Samsung Biologics. Established in 2011 and headquartered in Sangdo, South Korea, Samsung Biologics is a fully integrated CDMO offering state-of-the-art contract development, manufacturing, and lab testing services. Today's broadcast will learn about approaches to demonstrate process understanding needed to develop a robust and scalable manufacturing process to support successful GMP manufacture of biologics. And to get us there is going to be Kenneth Green and Thomas Gervais. Kenneth Green is currently Senior Director of the Manufacturing Science and Technology team at Samsung Biologics, following previous roles at the company in cell culture manufacturing and DS operations. He has prior experience in process development and characterization, technology transfer, validation, and manufacturing, including leadership roles in process development, MSAT, and tech services with Lonza, GSK, Amgen, Pfizer, and Shire. After Ken, we'll hear from Tom Gervais. He's a Director of Manufacturing Science and Technology team, Downstream at Samsung Biologics. His prior experience includes biologics process development and characterization, tech transfer, and commercial manufacturing, including leadership roles in process development, MSAT, and process lifecycle management with Cambrix Bioscience, Centocor, and Bristol Myers Squibb. We will be accepting questions throughout the talk, and you can send those in using the box at the lower left of your screen. Questions will be answered after the presentation in a brief Q&A, and this presentation will be archived on our website, contractpharma.com, for 1 year. If you're experiencing any kind of tech difficulties, you can type those into the box as well, and someone from the broadcast vendor will help you out. So that's about it, and if you're ready, Ken, it's all yours.
Okay, thank you. This is Ken Green. I'm joined by my colleague Tom Gervais. We're part of the MSAT organization at Samsung Biologics. What we'd like to talk to you today is really our approach to process development and characterization and how we're able to blend both small scale and manufacturing scale data and know-how together to really give us a full picture or understanding around our processes. So I'm going to start by a brief introduction to Samsung Biologics. Tom will take us through our approaches for process characterization. And then we're going to touch on a few case studies. So we have 3 case studies lined up with real examples, manufacturing examples, to talk about how we've kind of blended, as I mentioned, at scale manufacturing data with our small scale understanding. And finally, we'll leave some time for questions and answers. Okay, Samsung Biologics started in 2011, and since that time, we've grown now to having over 2,600 employees. We've developed 3 drug substance manufacturing plants. We have a total capacity now of 364,000 liters. We have a great success rate in terms of batch success, over 95%. And since we've had our first approval in around 2015 or so, we've had over 50 approvals, with 30+ inspections, and now working with over 40 different global clients. A quick overview of our manufacturing facilities. So as I mentioned, we started in 2011, built our first drug substance plant starting then. This was commissioned in 2013. The first plant had a capacity of 30,000 liters. This was quickly followed by Plant 2, started in 2013 and fully completed in 2016. This added an additional 154,000 liters cell culture capacity, including 10 15,000 liter bioreactors, and then 2 additional suites, small scale suites of 1,000 liters each. And then finally, we've recently added our third plant, which was commissioned in 2018. Which added a further 180,000 liters of cell culture capacity. In addition to the drug substance, we also added drug product capability, fill finish capability over the years, and coupled with that, also we developed quality control labs and process development labs to support the manufacturing sites. The facilities have all been designed to operate as multi-product facilities with a very detailed focus on flexibility and efficiency. So over the years, we really developed innovative approaches to really maximize our production cadence, really focusing on things like minimizing product changeover, innovative approaches to preventative maintenance so that we have a slowdown as opposed to a shutdown for maintenance. And we've really been able to kind of ramp up our production cadence and minimize downtime. In addition, although we have very large-scale equipment and plants, we also use a lot of single-use parts and equipment. So we'll be able to blend stainless steel and single-use together. So that's been a feature to allow us for additional flexibility. In addition to the manufacturing, we've also developed, I would say, innovative approaches for validation for both facility equipment and process validation. And then in terms of how we manage our vendors and our supply chains, as well as technology transfer. In addition, then we also have full laboratory services, and this is what Tom and I will talk about, including full capabilities for process development, process characterization, and analytical testing. With that, I'll hand over to Tom, and he'll talk through our development and characterization capabilities.
Thank you, Ken. So as Ken said, I'll describe our process development, characterization, laboratory capabilities. Firstly, I'd like to speak to our upstream process development and laboratory capabilities. This is an overview and of the responsibilities that we have and capabilities that we have. So firstly, we have capability for basal and feed media screening using commercially available media products. We can also accomplish process parameter screening and optimization based on design of experiments using our mini bioreactor systems. We have capabilities with the Amber 250 systems and 2-liter bioreactor systems. We can conduct process verification for when we receive processes from clients based on final media formulation and defined process parameter ranges. And these verification runs start with thawing a cell vial, expanding through shake flasks and other systems, up to 2-liter or 10-liter bioreactor systems. And finally, we can produce clinical material at clinical scale productions up to 1,000 or 15,000 liters, depending on what the process requirements are, or client requirements are. And while we're doing these runs at manufacturing scale, we also will perform satellite runs in the lab in parallel to monitor the process. From the downstream perspective, we also have laboratory capabilities that mirror the upstream capabilities. We can conduct chromatography process development for protein A steps or any other type of modality of chromatography, ion exchange or hydrophobic interaction. We can conduct or have conducted filter process development and filter sizing for normal flow and tangential flow filtration with the various systems that we have on site. We also conduct verification runs to confirm process robustness and reproducibility. We do this with our verification runs, taking the materials from the 10-liter scale or up to 10-liter scale bioreactors from our upstream colleagues, and to run those processes in that sense. And lastly, we support clinical scale production and also perform satellite runs in parallel to demonstrate process consistency and capability. So we also have analytical laboratory services and capabilities. We can conduct analytical method development for liquid chromatography, capillary electrophoresis, immunoassays. We also provide analytical support for the measurement of amino acids and other media compounds, or in-process analysis for upstream or downstream development work. Extended characterization and comparability studies are also a service that we provide for structural characterization, mass spectrometry, chromatography, immunoassays, and biological activity assays. And we also provide manufacturing support for composition analysis, particle investigation, leachables and extractables work, And our deliverables will be reports, or mostly method development reports, technical reports, and analysis reports. So now I'd like to switch gears a bit, or provide an overview of our process characterization study flow, kind of our basis for this. Starting in the left-hand bottom corner, where we will receive historical information on the product and the process from our clients. With that, we will evaluate the CQA list and critical quality attribute list and acceptable ranges that are known at that time from the client. And then we'll establish a process characterization response limits based on all that historical data. And then we'll move into a risk assessment of failure mode and effects analysis to then begin thinking about how to set up the process characterization. While we're doing this, we'll create scale-down models so that we have appropriate qualified scale-down models for our process characterization work, either at the 250 milliliter scale, 2-liter, or 10-liter bioreactor scales. And then we'll move into creating a process characterization plan based on all the historical data in our FMEA to set up a process characterization study design, either using a working cell bank or master cell bank, depending on the needs and the timing of the project. The first set in the process characterization design will be a screening DOE, usually greater than 5 parameters, to establish appropriate parameters to look at more clearly or more closely. For the second step, which is the response surface model DOE, which is approximately greater than 3 parameters. After that, those data are reviewed, then we will look at a third set of OFATs, one factor at a time studies, to look at any parts of the space that we hadn't looked at previously, or just to confirm some things. And lastly, maybe a set of worst-case studies to challenge the edges of the process itself, or the boundaries, as it were. And after that, we will summarize all this into a process characterization report. And in this report, we have a classification of the parameters themselves and establishment of acceptable ranges. So to summarize the process characterization and execution in summary, so firstly we'll start with the master protocol that contains the parameters we're going to look at and the limits that we're looking to explore. That follows on with a screening study for the selected parameters determined by the FMEA. A statistical analysis of those study results will then occur after the screening DOE, which will then follow into the response surface modeling DOE. Once those data are generated, we'll analyze the data and then follow that up with a OFAT and worst case study set of studies. And then all the results will be compiled, analyzed, and the results will be provided in a process characterization report, where the parameters themselves will be classified as either critical, key, or non-key, depending on the responses and how they relate back to critical quality attributes. And then we'll establish PARs for the CPPs, KPPs, as they come. So all this work that we've done for process characterization and some work that we haven't done for process characterization, but have allowed us to do tech transfer of client projects and scale up, either from a client scale of 2 liters to 5,000 liters, scale down from 12,000 to 20,000 liters, or scale to scale, 5,000 or 15,000 liter scales coming from the clients. So we've been very successful in taking our clients' process scales, whatever the scales are, utilizing our gap assessments and our verification at 10-liter scale, and then transferring that and appropriately manufacturing that in our 1, 5, or 15,000 liter manufacturing scales. So that concludes our approach to process characterization and our tech transfer that we've accomplished using our laboratory capabilities. Now we'll move into our case studies that Ken had mentioned previously. And so I think, Ken, is there a case study that you have in mind where we've developed something in the lab and then implemented that in production?
Yes, thanks, Tom. So the first case study I would like to go over is a new application for us where we've implemented a perfusion system for what we call our N-1 bioreactor. Our N-1 bioreactor is our final seed bioreactor before we inoculate one of two production bioreactors. So I want to talk a little bit about our approach, how we developed that, how we implemented, and then ultimately how we verified that with our small-scale model. So let me give you a little bit of introduction to perfusion and how it's applied to our N-1 bioreactor stage. So perfusion really allows us to achieve higher cell densities really by removing spent media, waste metabolites out of the cell culture and replenishing that with fresh medium. To achieve that, what we do actually is we remove the cells from the bioreactor using a perfusion device. The spent media is filtered out. And then as that spent media is filtered out, we add fresh medium to the bioreactor vessel at the same rate. And by doing that, then we can maintain the cells in an active growth phase, obviously once the cells are returned back into the bioreactor. So we do this with a perfusion device called ATF, which stands for Alternating Tangential Flow. And we applied this to our 3,000-liter N-1 seed bioreactors. As I mentioned, the benefits of this really then we can grow our cells to much higher cell densities. And by doing that, then it has benefits for our production stage where we can seed at high densities and achieve much higher production in our production phase in a shorter duration. So let me tell you a little bit how we configured this in terms of our bioreactor trains. On the right side of this graph, of this slide, you can see the scale up in our bioreactors from left to right. The smallest vessel is 120 liters, and then we have a five-fold scale up to 600 liters, and then to 3,000 liters. It's the 3,000-liter stage where we've applied the ATF perfusion. And then that seed bioreactor will feed one of two production bioreactors at 15,000 liters. Again, as I mentioned earlier, the schematic on the left just shows a description of what I talked about, how the cells are removed from the bioreactor. They're retained within the ATF. Spent media goes to waste, and then the cells are returned the action of a diaphragm pump. And at the same time, then we're replenishing the media by adding fresh feed media. So I just want to talk now a little bit about how we went about designing this, and a little bit how we qualified this, and a little bit of data from GMP manufacturing. So part of the design here was obviously we're running this N-1 seed bioreactor in a different state. We are having much higher cell densities with much higher oxygen requirements. So the first step around this was really to do some process simulation to really understand the oxygen requirements, typically through aeration and agitation, to really see if we could understand that, so that would support the growth requirements of the cells. The path for this, obviously, we spent a lot of time in the lab using our small-scale models, working on both lab-scale models and process design. Once we were confident with that, we went into a testing phase, and then qualification and validation of the system, and finally into GMP operation, where we verified the at-scale performance with our small-scale model. So the first part of this was really, again, looking at our small-scale model and making sure that it was truly representative of a manufacturing scale vessel. So again, what we did here was really try to make sure that we can mimic our cell densities, our cell viabilities, but using our 2 and 10 liter small-scale models, with our 3,000 liter data. And again, what we've got is really very good comparison where we could achieve the same types of cell densities with the same viabilities with our small scale, and got good confidence with that model. The second part of this was then trying to figure out the scale-up strategy. As I mentioned earlier, the cells, because of the higher densities, will have a higher requirement for oxygen. So we wanted to make sure that the design of the N-1 seed bioreactor could provide sufficient oxygen. So we did some work again, looking at process simulations on things like spargy design, gas in flow rates. And then in addition to that, thinking a little bit about, okay, well, what's the potential for shear stress on the cells? What is the potential for accumulation of dissolved carbon dioxide? An ability for us to strip that out so it didn't become inhibitory for the cells. So we spent a lot of time really focusing on the scale up, what are the important parameters, and then obviously trying to mimic that at small scale. This shows a little bit of data on the at-scale data. On the left side of this, you can see the results from our N-1 perfusion. This is a 3,000 liter. Again, what we achieved was the desired cell densities. Our maximum cell densities was 9 times 10 to the 7 per mil, which is approximately 10-fold more than a typical batch bioreactor. So we were able to achieve a high cell densities. The viabilities again remain very high. As I mentioned earlier, we're actually removing cells from a bioreactor into the perfusion device and then returning them. So for us to be able to maintain high viabilities throughout the course of the culture was really important. And then the graphs on the right show our actual production culture, which have been fed from our N-1 perfusion. And what it really shows, if you're familiar with growth curves in a production bioreactor, you normally get an initial lag phase. The cells will go into exponential growth, reach peak, and then decline. What we found is we could inoculate the production bioreactor at a much higher cell density. We didn't get the lag phase. The cells were pretty much right into exponential growth phase. And they hit peak viable cell densities at a much earlier stage than a typical batch or fed batch culture. At the same time, again, if you look at a viability profile from the production stage, it followed a typical pattern in terms of a slight decrease over time until the harvest time. So this system, again, was very successfully scaled up. As I mentioned, it was a new capability for our Plant 3, our new facility. Again, we were able to cultivate much higher cell densities, up to 10-fold compared to traditional seed culture. I talked a little bit about the benefits for the production bioreactor, being able to achieve higher cell densities, higher productivity, and within a shorter duration. And again, I think this has been very successful for us, and we were able to, again, qualify, validate, and get into GMP production within a 6-month window from the design of the ATF. So a very successful project for us. Okay, next I'd like to switch gears, and Tom is going to give us a different example, a different case study, but again, where we've used, I think, both at-scale understanding with our small-scale models to determine a solution. Okay, Tom.
Thank you, Ken. So this next case study will involve lab-scale verification work and satellite run work in which we arrived at a solution for basically an unusual investigation that we had. For verification and satellite runs, we typically do these during tech transfer. They're standard for our component in our MSAT group here at Samsung Biologics. And not all processes or not all process understanding is captured in the client documents. We found that over our experiences here. And we've decided, and what we can determine from executing the verification runs, is that it demonstrates that us as the receiving site can execute the process as intended. It also helps us reduce risks due to gaps in the information and helps us to evaluate process changes for facility fit. And lastly, it helps us identify any potential manufacturing issues that could possibly occur, since we've actually run it at the lab scale first to really understand the process. So, as I said, satellite runs, we run these during the manufacturing scale, or during manufacturing to confirm results. In the schematic here at the bottom, we have a generic manufacturing timeline. Our verification and satellite runs are in the green arrows. We have the options or opportunities for any supplementary runs that we would like to do in the gray arrows. And you can see that the manufacturing are shown in the blue arrows, the engineering run and PPQ runs. And you can see that satellite runs coincide with those engineering and PPQ runs themselves. So our case study here has to do with an unexpected process issue during the engineering run that led to delays and product loss. So what we had experienced was clogging the in-process harvest filter. We had an unexpected pressure increase to greater or up to 9.9 bar. And this clogging actually then continued on to the protein A guard filters, where we ended up using about 18 filters per cycle for a total of 54 for 3 cycles, where the expectation was around 7 filters per cycle. And so that was definitely concerning. And subsequently, we recognized in the second column in the purification process that we had approximately greater than 20% loss, 25% loss in the second column step, 20% loss of yield in the first column step. And we actually evaluated materials and found that there were high levels of half antibody and HMW in the third column step effluent. So looking at our satellite runs for confirmation, so we had our verification run from the beginning of the project shown here as the blue chromatogram. And you can see that the satellite chromatogram, which was the materials taken directly from manufacturing and run in the laboratory, in the pink chromatogram, that the chromatograms don't match. And this particular set of chromatograms is from the second column step. And so we had an early indication of that something was wrong with how the process was running via the chromatograms themselves. And then taking samples from the satellite run and the engineering run, and then and also we had data from the verification run. So if you look at the top graph, or top figure, on the right, we show half antibody. The blue bars are the satellite runs. The gray bars are from the engineering run. And the orange bars are from the verification run. You can see for the first column step, and second column step, and third column step, the verification runs shown in the orange had half antibody of around 3%. For the engineering run from manufacturing, we had 46.9%, and then 35% for the second column, 32% for the third column. The satellite run had 6% for the first column. The second and third columns mimicked the engineering at scale runs with 35 and 31% respectively. The graph on the lower right is the HMW graph, and we see similar behavior with the verification run having 1 and less than 1% of HMW for first, second, and third columns. Engineering run had 25, 26%. Column 2, the second column had around 18%, and the third column around 18% HMW. And the satellite run for the first column had 4%, but for the second and third columns, 19 and 18% respectively. So our satellite run data was giving us an indication that the engineering run was also having the same problem. So the investigation into this, so the satellite run suggested that that there was a quality issue, and it turns out that we looked at some operational execution to see if there were any errors done there. So the raw materials in the downstream process were found to be in good quality. The buffers were prepared correctly. The equipment status, all the equipment used in downstream were of good working order. We evaluated if operators were following procedures, and we found no significant errors in any of the procedures and any of the MBRs. And lastly, we looked at hold times and transfer durations, and we saw that there's nothing out of expectation. So operational error, execution errors were ruled out as the issue there. So since no root cause was identified in the downstream process, we started looking at the upstream process, going from cell culture prior to the first column step. And what we had investigated and determined is that there was a reduction in the antibody due to an unanticipated enzymatic activity, whereas the high HMW and the high half antibody could be attributed to this enzymatic activity and then subsequent exposure to low pH. So then we determined that, okay, what potential mitigation actions should be evaluated for the next manufacturing batch. And the three things that were looked at were to perform purification at reduced temperatures at 5 degrees C to reduce the enzymatic activity, control dissolved oxygen at the harvest to prevent reducing conditions that would enable the enzyme to cleave the antibody, and also perform the proteinase step as fast as possible so that we could separate the antibody from the enzyme, thus minimizing enzyme contact time and activity. So to summarize, our verification runs, they help reduce risk prior to large-scale manufacturing. They also establish a baseline of what one should expect for the process, and also establish a control for the process in the event that we have to do an investigation, such as was the case in this instance. Our satellite runs are definitely useful for troubleshooting and provide an early look at product quality. They enabled us to look at the product quality in this specific issue and also see the impact of this major process challenge. So realizing the benefits of the verification and satellite runs requires strong technical technical foundation and understanding. And this is critical for clients who select a CMO who offer the right balance of technical capability and expertise. And so the needs differ amongst clients, whether it be a large biopharma or a virtual biotech, but a CMO that has the right balance of technical capability and expertise is definitely required. So with that, I'll turn it back over to Ken, and I think he has a particular case study where we were doing characterization of a process step that unfortunately we couldn't really utilize the laboratory to really understand this process step.
Okay, thank you, Tom. So our last case study is really around our harvest step. And this is an interesting one because it's an operation really we can't mimic at small scale in the lab because of the nature of the equipment. So I'll talk a little bit about how we characterize this at scale, but also supplement our understanding with some small scale studies. So the harvest operation itself is really when we take the cells from the production bioreactor and we run it through a centrifuge to remove cells, and then through a series of filtration to remove cell debris. So the area we can't really mimic in the lab is for centrifugation. But what we are able to do is to assess that at scale by looking at a number of study parameters and getting some at-scale data to really understand performance. So I'll talk a little bit about that, and then obviously downstream of that then we have flexibility with how we design our filtration. We typically start with some lenticular type filters, coarse filters to remove the larger particles, and then typically some membrane filters which will be bioburden reduction filters as polishing filters prior to collection in a harvest tank. After collection in the harvest tank, then the process stream will go to purification. So when we look particularly at the centrifuge itself, we follow a kind of an industry approach around we design the scale up. Typically we use Q over sigma, where Q is the flow rate and sigma is the sedimentation area. And we really look at two parameters to understand performance, notably bowl speed and flow rate. And as I mentioned earlier, what we will do is typically perform a study at scale where we will purposefully change conditions within a design space, such we would select best and worst case conditions as well as a center point and run the centrifuge for a period of time under those conditions to really assess performance. Now, from a performance perspective, really what we're looking for is separation efficiency. So we're really looking at the output of the centrifuge in terms of centrate clarity. So we look at the particulates. So we measure obviously the particulate level, the particle size distribution. We then go into the lab and do filterability studies with small scale filters to understand the filter characteristics of the centrate. We typically look at other impurity analysis, for example, host cell proteins, host cell DNA. We'll measure that to see that we're getting either clearance when it's claimed or we're not increasing the level of host cell impurities. One of the concerns, obviously, when you introduce a centrifuge to the process is the potential for introducing shear and having an increase in things like host cell impurities. So we'll take a look at those again during our at-scale study. And also we'll look at LDH, lactate dehydrogenase, which will be an indication of cell shear. So as I mentioned, we will run a study at scale, but we also use our manufacturing experience. So we've done this type of study many times for many different types of products. So we know instinctively where a good operating space is, and really this exercise at scale becomes more of an optimization. And as I said, we'll pick conditions corresponding to best case, worst case, and center point, depending on the bowl speed and flow rate. Downstream of the centrifuge, then we have the filtration. So again, what we'll do in the lab is look at different filter options, different filter types, filter sizings, look at things like flow rate and pressure curves to really understand the best filtration combination. And again, that's all work we will do at the lab, but we will take samples from the at-scale manufacturing from the centrifuge to perform those types of studies. And this is kind of always interesting for us when we work with a lot of our smaller clients who are really operating at smaller volumes, where they don't necessarily use a centrifuge for cell separation. They normally would be able to go from a production bioreactor straight into a depth filter and do the clarification that way. And what we found is once you scale up, typically above 5,000 liters, then you really require a centrifuge again for cell separation. The downside, if you don't use a centrifuge, is that you end up with an enormous amount of filtration area to be able to clarify the stream. So again, we have some, based on our experience, we have recommendations, as I mentioned, on not only the centrifugation conditions, but also the downstream filtration. Okay, and then with that, then I think I'll turn it back over to Tom, and Tom, you can go over our conclusions.
Yeah, thank you, Ken. So in conclusion, we've taken the time or the opportunity to discuss with you some of the things that we've done here at Samsung Biologics to leverage our manufacturing experience to accelerate process characterization and validation. We've used our qualified small-scale models to represent manufacturing operations from 1,000 liter up to 15,000 liter scale. We have a dedicated and experienced group of MSAT personnel who have experience with over over 40 product transfers that have been successfully completed to date. We have state-of-the-art laboratories for upstream, downstream, and analytical, and we have capabilities to successfully do the transfer work and characterization work here. And we've described some of our solutions and developed solutions for high viable cell densities, high titer processes, and also for MABs that have sensitivity to process conditions such as shear stress or disulfide bond reduction. And lastly, I think we want to end with our virtual, or an ad, I guess I could say, for our virtual exhibition hall. So please visit our virtual exhibition hall. This launched this week and at exhibition.samsungbiologics.com. And so you can visit there to learn more about our service offerings and unique strengths. And with that, we'll take any questions.
All right, great. Thanks a lot, Tom and Ken. Great presentation, guys. And that virtual booth looks pretty cool. Starting to see more of those pop up these days. All right, let's see. We've got a couple questions to get through here. Let's start with first up. Have you scaled up upstream fermentation after screening and optimization at 50 liters or similar scale? And if yes, any challenges?
Yeah, I mean, it's again, our typical approach would be to scale up from the bench for lab scale. We have, small scale models at 2 and 10 liters, and even actually smaller systems we scale up from. And we also have some pilot capabilities. So yeah, most of our all our processes, we will develop a small scale model and verify that in the lab prior to scale up, which is part of our normal or routine tech transfer process. And we have a high degree of confidence with our small scale models, particularly our 10-liter model. Will scale up very effectively to all our plants. So that's ranging from 1,000 to 15,000 liter scale.
Okay, great. Let's see. Next question asks, is cumulative hold time part of a worst case scenario?
Yeah, it could be. I know that many studies have been done to look at cumulative hold time and looking at over the downstream conditions. So yes, it is part of worst case studies. One could add them. Well, usually one adds them later, but the short answer, yes.
Yeah. Okay.
Yeah, I think just to add to that, I mean, what we would typically do for in-process hold studies is really a combination, again, of at-scale hold conditions versus small scale. So again, it's using, again, qualified models at small scale to demonstrate we can hold under representative conditions at the manufacturing scale and use a combination of scale data.
Yeah. Okay, great. Let's see here. What is next? At what stage of a project should one start process characterization?
Well, I guess it's kind of a risk-benefit thing in a certain sense. So you can start process characterization anytime you want, but it's definitely it's an intensive amount of work and a lot of effort goes into it. So most clients tend to wait till their product gets a little bit more mature within the clinical space. Before they embark on process characterization, but you could start it anytime you want.
Yeah, I think typically from our practice, most of our clients would really start at phase 3. I think that's when they would consider a process lock, and I think that's when they would go into a full-scale process characterization. But as I said, as Tom mentioned, the process understanding starts before then. So obviously, as we develop early clinical programs, we're gathering understanding. But I think from a formal characterization, that would be typically at around phase 3.
Okay. And did you have to perform virus clearance studies at 5 degrees Celsius?
I think a few of our clients have. Again, it depends on the molecule and what you're trying to accomplish, but you can do them at 5 degrees C. I don't know of any client that was specifically was forced to do it. So I guess again, short answer is yes, you can do them, and some have done them, but yeah.
Yeah, it really depends, I guess,
where
the process is run and whether it's run at ambient versus cold conditions. But but we do have cases where we run under cold conditions, and I think the viral clearance will be done under similar conditions.
Yes, indeed, that's correct. Can.
Okay, gotcha. And are satellite runs useful in addressing scale-up related issues?
Oh, definitely. So actually, a great way to get understanding for scale-up issues, if you see, let's just say, aberrant behavior is what the case that I presented, then you can really start looking at it from, is this a scale-up issue or not? And, so I would say yes.
Yeah, okay. The nice thing about a small-scale model or a satellite is that you can rule things out very quickly, which are maybe not related to scale or not related to process. So again, I think for us, it's really critical to run those so we can really understand are we representing what we're seeing at scale, and in terms of an investigation, we can quickly do some diagnosis based on that small scale data.
Okay. Yeah, I agree with Ken. It's a good tool to have to do the investigations and to rule things out.
Okay. Let's see. How would you share data and info in development as well as in clinical and commercial manufacturing?
Again, I think we are very transparent with the data we generate. I mean, it's really all our clients' data. So essentially, we will share everything that we generate, both in development as well as in our manufacturing. And again, we've developed systems where we can share that data in I would say close to real time, but really having the client having access to that information as quickly as possible. And we will share, again, whatever the client requests in terms of certainly process information. Anything to add there, Tom?
No, yeah, it's the client's data, so we'll share everything.
Yeah.
Okay.
I think where we can add value is really our experience with maybe other programs where we have some best practices or kind of some more generic solutions based on our experience. I think that's where we start to add value in terms of interpreting data and understanding again how process development and characterization really translates to at-scale manufacturing.
Okay, great. Let's see, time here for one or two more. What is the total time spent during process characterization?
So yeah, our model is typically 12 to 15 months of that order. It's really hard to kind of shortcut process characterization just because the number and types of studies we have to perform. But it's typically, it would be I would say 12 to 15 months is kind of our standard timeline.
Okay. Okay, great. Let's see. Let's get one more in here before we pull the plug. What are the most crucial precautions to be taken for manufacturing monoclonal antibodies in a bioreactor?
Yeah, I mean, I think for me, it's the typical things around understanding, if there's any sensitivity to shear, so agitation, gassing rates, sensitivity to foaming, and then again, I think just understanding the variability that you may have with the raw materials, whether it's the basal media or the feed media, and understanding, that there may be some variability associated with that. But again, it's fundamentals around it. It's really around oxygen mass transfer, being able to strip PCO2 so it's not inhibitory, and then just making sure, again, things like osmolality are controlled.
Maintaining cell health.
Okay.
Really...
Okay, great. So we've had more questions come in. If we didn't get to your question, Ken or Tom, they'll follow up with you directly. So thank you both Ken and Tom for a great presentation. Thanks again, Samsung Biologics, for your sponsorship, and everyone in the audience for attending today's session. As I mentioned earlier, this session will be archived on contractpharma.com for 1 year. Thanks for joining us and have a great day.